Hi, this is HYBRIDIZATION and I am going to present my video on the topic microtechnology, and my case specific topic is Drought induced gene expression analysis in Arabidopsis and related plant species.
Microarray hybridization is the multiplex technology most widely used in the global transcript profiling. It is evolved from southern blotting. Microarray technology consists of series of microdots of DNA oligonucleotides arranged on a solid support. These DNA oligonucleotides are also known as probe. Solid support is usually made up of silicon slide, glass chip, or microscopic slides. Then a fluorescent RNA or cDNA is hybridized to the slide and intensity of fluorescence is measured and analyzed. There are various types of microarray are used, such as DNA microarray, protein microarray, and tissue microarray.
Coming to the point of microarray technology, microarray is also known as the DNA chip of the biochip, in which the one silica plate or the glass plate is used on which the array of a DNA or the complete genome sequence of a particular rice is imposed, and it is used for the very purpose in case of the microarray technology.
Microarray consists of number 1, gene chip feeding station 450, number 2, barcode reader connected to the computer, number 3, hybridization oven 645, number 4, alphametric gene scanner, and finally microarray chip.
Microarray technology is the hybridization, in which the nucleic acid, that is the DNA or RNA, is complementary binding to the probes which are affixed on the microarray chip. Here is the gene chip hybridization of oven 645 chamber. Here is the hybridization unit. In this, those of the chip which are applied with RNA solution is kept under for the hybridization procedure. Here, this is the position where we can keep the microarray chip. 8 microarray chip can be kept at the same time, and then along with the hybridization buffer we are keeping the solution inside the chamber, and at a particular time we will allow it to run.
RNA isolation by triazole method is a simple and efficient method. Here, triazole kit consists of DPC water 1 ppm, triazole for integrity of RNA, 75% ethanol act as a salt remover, 2 molar NaCl isopropanol for RNA precipitation, chloroform as forming an organic phase, a set of pipette man and tips, ice box, and last discard box. First step is the crushing of 200 mg of Arabidopsis sample in liquid nitrogen and triazole reagent. Triazole reagent is added 500 microliter and crushed well. Then it is transferred to sterile Eppendorf tube and kept at room temperature for 10 minutes. Then allow to centrifuge at 10,000 rpm for 10 minutes at 4 degrees Celsius. After centrifugation, transfer the supernatant into another Eppendorf tube containing 0.2 ml of chloroform. Similarly, follow the procedure to transfer the supernatant of stretched Arabidopsis sample to the chloroform Eppendorf tube. Then incubate the tube at room temperature for 5 to 10 minutes and allow to centrifugation at 10,000 rpm at 4 degrees Celsius. Then take out the tube from centrifuge and collect the supernatant and add 0.2 ml of isopropanol, 0.2 ml NaCl and incubate this tube at a room temperature for 1 hour. Again follow the centrifugation at the given condition. Then wash the pellet with a 75% DEPC treated ethanol. Here the ethanol acts as a salt remover. Due to the DEPC treatment, it doesn't affect the RNA. And at last, discard the supernatant and dissolve the RNA pellet in a DEPC treated water. Two tubes containing RNA pellet are obtained at the last of RNA isolation step. Here we can see the left side is containing controlled sample of Arabidopsis. This animation explains the centrifugation of Eppendorf tube containing Arabidopsis leaf sample. The cells are in the pellets. Then remove the liquid but not the cell with the help of pipette man. Eppendorf tube will contain only the pellets. From that pellet, we are going to extract the mRNA with the help of mRNA extraction buffer. Now, RNA is floating in the extraction buffer. Then, remove the RNA and place it in fresh tube. Here, we are going to prepare a cDNA with the help of reverse transcriptase enzyme. It is labeled with the two different dye, Cy3 and Cy5. The Cy5 is containing red colored and Cy3 is indicating green colored, that is a controlled DNA sample. Then combine the red and green cDNA in a single one Eppendorf tube. Now we will go to see the DNA chip. This is the microarray chip which is consist of 60 to 70,000 spots. Each spot represents the DNA or probe. Now let's we take randomly any 3 spots corresponding to 3 genes. Now we are going to add the cDNA samples on a spot with the help of pipette mat. It will bind to the probes complementarily. The labeled cDNA has bound to the spot. The magnifying image is showing the complementary bonding between the probes and cDNA sample. Out the unbound sample. Now what we see actually in the condition. We will allow to run the microscopic with the laser technology. Here, the 2 lasers are used to screen the spot. It will give 3 different kind of images: red colored, green colored, and yellow colored. Microarray chip scan image indicates green colored spot representing control DNA, red spot represents sample DNA, yellow color represents combination of control DNA and Drought DNA. Some of the black spots represent neither control and sample DNA. After the data visualization, we will count the red colored spot and green colored spot. From that, we will obtain the logarithmic ratio of psi5 to the psi3. It will give an idea of those of the genes which are underexpressed and overexpressed during the drought condition. Now, let's see the results and conclusion. Drought stress induced gene expression on profiling study includes 300 Arabidopsis genes. There were 44 drought-induced mRNA identified, out of which 30 mRNAs are novel. 12 of the stress-induced genes are identified as being regulated by DRIP1A, out of which 6 are novel. In addition to that, 11 of which were shown to contain dehydration responsive element, that is DRE, in the promoter region. Applications of microarray technology. Microarray technology is used in the field of mRNA and gene expression profiling, comparative genomic hybridization, chromatin immunoprecipitation on chip, and finally stealing array and exon generation array. It is also used in the advanced field like pharmacogenomics to study the effect of specific drugs and diseases, to study the epigenetic landscape and transcription landscape. Microarray is mostly used in SNP detection and alternative splicing detection.